Hello, you're welcome to our lecture series on biological techniques. We have been discussing about microscopy. Earlier in our discussion of microscopy, we had classified microscopes to various major types based on the source of illumination: the optical microscope, the electron microscope, the X-ray microscope, the scanning acoustic microscope, and many others. We had looked at the optical microscope that uses light and the various types of the optical or light microscopes, such as the bright field microscope, the dark field microscope, the uh, the inverted microscope, the fluorescence, and others. Then we also looked at the electron microscope that uses electrons to produce magnified image. Then we looked at the two types of electron microscopes: the transmission electron microscopes and the scanning electron microscopes. We also want to talk today about the X-ray microscope and others. Now, what is the X-ray microscope? The X-ray microscopes use a beam of X-rays to create an image. Now, due to the small wavelength, the image resolution of the X-ray microscopes are higher than in optical microscopes. The resolution of the images in X-ray microscopes are higher than the optical microscopes. The maximum useful magnification is therefore also higher and is between the optical microscopes and electron microscopes. So the resolution of the X-ray microscopes are also higher than those of the optical microscopes, and they're in between the electron microscopes and the optical microscope. So the resolution of the electron microscope is higher than the optical microscopes, but lesser than that of the electron microscopes. Now here is a picture of an X-ray microscope, a microscope that makes use of X-rays. Here you can see the eyepiece just as it is, with the optical microscope, the stage, then the fine and coarse adjustment knobs, and the objective, and other features that are similar to the optical microscope. Now, what's the X-ray microscope used for in biology? Three-dimensional X-ray microscope has proved invaluable in the field of medicine, and it has application in developmental biology, imaging of soft tissues, biomaterials, in vivo imaging, among others. The X-ray microscope is useful in the field of medicine, but in biological sciences, it is very important in the imaging of soft tissues and biomaterials, and it's also good in vivo imaging. So those are some of the applications of the X-ray microscope. It has been used to visualize the structure of various tissues during development. Then biopsy samples in model organisms and humans. So it is used in the study or to visualize various structures during development. Then biopsy samples and model organisms and humans. The advantage of using X-ray microscopy is that it can be used to analyze thick samples with great resolution. That's one very advantage of the X-ray microscope. It can be used to analyze thick samples, samples that are very thick, unlike the light microscope that will need to work on thin samples or microscopic organisms. This X-ray microscope can be used to work on thick samples, okay, with great resolution. The X-ray microscope has these very important features. Now I want to look at one important type of microscope too, the microscope that uses sound waves. That's called the scanning acoustic microscope. The scanning acoustic microscope. Now what's the scanning acoustic microscope? Scanning acoustic microscopy uses focused sound waves to generate an image, a process called scanning acoustic tomography. So instead of using light, instead of using electrons, instead of using X-rays, this scanning acoustic microscope utilizes sound waves to generate an image. And that process is called scanning acoustic tomography. It is an ultra-high frequency pulse echo ultrasonic imaging system that is used to look inside optically opaque solid samples and view internal structures. So it is very useful in viewing the internal structures of optically opaque solid samples. Optically opaque solid samples. So it's very important too in this aspect in viewing inside of optically opaque samples. Scanning each point individually with a wavelength of less than 1, the scanning acoustic microscope eliminates opportunities for interference and then creates the overall image by bringing together those obtained from each point. So it scans these organisms with a wavelength that's less than 1, and it creates an image that's very clear. Now here is a picture of the scanning acoustic microscope. Just like the optical microscope, it has an eyepiece, but it has imaging systems that is a monitor, a computer that helps to generate the images of the samples that are being viewed. What are the uses of the scanning acoustic microscopy or scanning acoustic microscopy? Scanning acoustic microscopy, or SAM, is the most current type of acoustic microscopy available to today's scientists. They can use it to view a sample internally without staining it or causing it to damage. So the sample can be viewed. The internal structure of a sample can be viewed without staining it, thanks to point focusing technology, which relies on a beam to scan and penetrate the specimen while it is in water. Now I want to look at another type of microscope that uses a different type of media or source of illumination to produce sound, and that's the helium microscope. The helium microscope. We want to look at the helium microscopes. We have 2 types of these helium microscopes. First, what are helium microscopes? Helium microscopes use helium atoms to generate an image. They use helium atoms to generate an image. There are 2 types of helium microscopes, and the 2 types of helium microscopes are the scanning helium ion microscope and the scanning helium microscope. Take note, the 2, one of them is the scanning helium ion microscope. There's ion in one. Then we have the scanning helium microscope. Now, we first want to look at the scanning helium ion microscope. That's abbreviated to be SHIM or HEIM. Now, what is a scanning helium ion microscope? As the name suggests, these devices use a beam of helium ions to generate an image. The scanning helium ion microscopes make use of a beam of helium ions to generate an image. There are several advantages to electron microscopes. One being that the sample is left mostly intact due to the low energy requirements, and that it provides a high resolution. So it has an advantage over the electron microscopes in that the sample is not destroyed, it is left intact, and it also provides a high resolution. The helium ion microscope opens new windows in nanoscale imaging. The microscope looks at the surface of objects by scanning the surface with a fine helium beam formed by a single atom at the tip of a 2-meter tall emitter. This produces images below 0.25 nanometer in scale, or the equivalent of 1 million times smaller than the size of a human hair. It is a relatively new technology, and the first commercial systems were released in 2007. This is a picture of the scanning helium ion microscope. A picture of the scanning helium ion microscope. Now, what's the scanning helium ion microscope used for? The current uses of the scanning helium ion microscope include ultra-high resolution imaging of biosamples, analysis of soot samples, that's smoke, black colored smoke or smoke from petroleum industry. Then studying of plant anatomy. The scanning helium ion microscope is also used to study the anatomy of plants. Then aerosol analysis, biomineralization studies, and mineral weathering studies. So it's used for a variety of studies. But what concerns biology most is the ultrasound imaging of biosamples and analysis of soot samples and studying of plant anatomy. Now I want to look at the scanning helium microscope, SHEM, the scanning helium microscope. The scanning helium microscope is a novel form of microscopy that uses low energy neutral helium atoms to image the surface of a sample without any damage to the sample caused by the imaging process. It uses low energy to produce low energy of helium atoms to produce imaging. And one very important feature is that the samples are not damaged in any form during the imaging processes. That's one advantage of the helium microscope, the scanning helium microscopes. Images are formed when a sample is made to raster underneath an atom beam, and by monitoring the flux of atoms that are scattered into a detector at each point. The technique is different from a scanning helium ion microscope, which uses charged helium ions that can cause a damage to a surface. This is the picture of the scanning helium microscope, a very sophisticated microscope that's used for various purposes. What is it used for? We want to look at the scanning helium microscopy is not yet fully commercialized, but it is seen as a complementary technology to existing microscopy with vast potential. Since helium is inert and neutral, it can be used to study delicate and insulating surfaces. It's a relatively new technology, a relatively new type of microscope, but it can be used to study delicate and insulating surfaces. It allows substances that are difficult to image, transparent, fragile, weakly bound or very high, very rough or magnetic to be analyzed in a non-destructive fashion. While the resolution of images is not as good as scanning electron microscopy, scanning helium microscope images have greater contrast, which makes better images in some applications. Now, thank you. I want to look at the neutron microscope. The neutron microscope is quite different from the helium microscope. Now, neutron microscope use neutrons to create images by nuclear fission of lithium-6 using small angle neutron scattering. Neutrons are much heavier than photons and electrons used in forming photographic or X-ray images. Now, what are some other features of the neutron microscope? First, before we look at that, here is a picture of the neutron microscope. What are the scanning probe microscope is the next type of microscope we want to talk about, the scanning probe microscopes. The scanning probe microscope forms images of surfaces using a physical probe or sharp probe that scans over the surface of a sample, gathering data that is used to generate the image or manipulate the atoms. Now here is the picture of the scanning microscope, the scanning probe microscope. Just like the optical microscopes, it has an eyepiece, okay, and the stage and other features. Uses of the scanning probe microscopy in biology. The primary benefit of scanning probe microscopy is its ability to generate a near physiological, to operate in near physiological conditions. The scanning probe microscope is able to operate in near physiological conditions. This enhances the performance of in situ laboratory analysis of biological and organic samples. It enhances the performance of in situ laboratory analysis of biological and organic samples. Researchers can consider the structure and properties of biomolecules, cells, and tissues close to native conditions, which has proven extremely useful in biological and biophysical research. Okay, the structure and properties of biomolecules, cells, and tissues are studied in close to native conditions, in their natural conditions. And this has been very useful in biological researches. Now, conclusion. We have discussed about various types of microscopes. Now, microscopes as we have discussed, can be classified based on physical principle that is used to generate an image. And as such, we had looked at the optical microscopes, the electron microscope, the scanning acoustic microscope, the neutron microscopes, the X-ray microscopes, and others. Different microscopes visualize different physical properties of the sample. Example, elasticity can be visualized with acoustic microscopes. Image contrasts, resolution, which determines magnification, and destructiveness of the sample are other relevant parameters when discussing about microscopy. Thanks a lot for watching this episode of our video. Keep watching our videos on YouTube, and please subscribe. Thanks.